Now you can say hello, sir. You can say hello now. He will listen.
Hello, Dr. Bapat. Good morning. Srinivasan here. Srinivasan.
Good morning, sir. Dr. Singh, you will have to keep the computer a little closer to sir.
Yeah.
Yeah, now it is fine.
Long time since we have met.
Yes, sir.
Bapat sir, I think whether his mic is unmuted or muted. I don't know whether he is listening. I think why she.
Karthik, if Vandana madam is there,
Sir,
there is an echo from your side.
Sir, we are waiting for Professor Karmalkar to join. Maybe in a couple of minutes he will join, then we can start the program. Sir.
Sure, we'll just wait for another 2 minutes and we'll start and then we'll contact him.
Sure, sir. What is it?
Yes. Very good morning to everybody. I'm audible?
Yeah.
Okay, good. Nice to see everybody joining in. So a request for most of the people, please keep your camera on so we can see your august presence and feel your presence online. And your microphones can be kept on mute. Later when you wish to interact, that time you can unmute it.
You say hello to them. Huh? Just mic testing. You say hello to them.
Hello. Am I audible?
Yes,
Yes,
sir,
sir.
perfectly audible. And happy to have you amongst us.
Thank you very much. So I'm equally delighted to see you.
It's so we are just waiting for Professor Karmalkar to join. And
We can start the program. He will join little later. Okay.
okay, yes, sir, great. So I wish everybody a very good morning and a hearty welcome to one and all on the occasion of the 75th Founders Day celebration at Agharkar Research Institute. Our Platinum Jubilee year also coincides with the celebration for the 75th Azadi Ka Amrut Mahotsav. Although COVID curtailed the gatherings, we have persisted and continued with our webinars, meetings, presentations in the online mode to mark the Platinum Jubilee celebration. Our chief guest, Professor Sandeep Verma, Secretary, Science and Engineering Research Board, DST, and guest of honor, Professor N.R. Kalmalkar, Vice Chancellor, Savitribai Phule Pune University, are gracing today's occasion. Without any further ado, I request our respected director, sir, Dr. P.K. Dhakefalkar, to deliver the director's report.
Are my slides visible?
Yes, they are. They are.
Yes.
Yes.
Thank you so much, Vandana. Good morning, everyone. Today's chief guest, respected Professor Sandeep Vermaji, guest of honor, Honorable Vice Chancellor of Pune University, Professor Nitin Karmalkar, respected life members and office bearers of MSCS, my fellow scientists, staff, and students. It is indeed a pleasure for me to present the report of the Agharkar Research Institute's contribution over the period of last year. Agharkar Research Institute of Maharashtra Association for Cultivation of Science was established in 1946. So it has completed 75 years of its existence. And it will indeed be a pleasure for us to take a small look back at this particular institute. Professor S.P. Agharkar or Shankar Purushottam Agharkar was a scientist, a renowned botanist at Indian Association for Cultivation of Science. After his retirement, he was thinking about starting a research institute where retired or retiring scientists could contribute significantly. At that time, there were stalwarts such as Professor Gharpure and Professor Jaikar, who was the founder vice chancellor of Pune University. They were also thinking about starting a research institute in Pune. When they came to know about intentions of Professor S.P. Agharkar, they approached him and immediately requested him to start the institute in Pune. And they offered him place in the basement of Law College. MSCS initially was registered as a society in October 1946 under the Society Registration Act of 1860 and later on also as a trust under Bombay Public Trust Act. It started attracting the scientists and academicians who actively contributed in various research areas in different sciences and the life sciences, biometry and nutrition, botany, chemistry, genetics and plant breeding, geology, etc. Several of these research programs were supported by the government of India as well as other private agencies as well. And because of their contribution and interaction with several government agencies and other funding agencies, there came into existence a vibrant research center which was known as MSCS Research Institute. Later on in 1992, to recognize the contribution of our founder director, the Research Institute was renamed as Agharkar Research Institute. In 1966, the Agharkar Research Institute, or then the MSCS Research Institute, was taken under the umbrella of Department of Science and Technology as an autonomous institute. And we are indeed in great debt, and we want to express our gratitude to Department of Science and Technology for their continuing support till date, and I'm sure in future as well. The institute was founded with a perfect mission, that is to conduct basic and applied research in life and related sciences for human betterment, to explore the genetic diversity of microbes, plants, and animals, and develop sustainable technologies for cleaner environment, agriculture, and betterment of health. The thematic research areas were identified, and over the years, these areas kept on getting improved, getting modified. And today, we have six thematic areas that have been identified, which include biodiversity and paleobiology, bioenergy, bioprospecting, developmental biology, genetics and plant breeding, and nanobiosciences. We have a strength of 32 against the sanctioned strength of 58 scientists and total staff strength of 120 against the sanctioned strength of 190. I would like to elaborate a little bit about the research that is being carried out under different thematic areas. The investigation, the first thematic area, it is about biodiversity. Now, under biodiversity thematic area, we investigate diversity of plants, microbes, bacteria, fungi, and also animals, microanimals in some cases. Now, the first area is documentation about biodiversity of novel taxa. The question that is asked is, why do we investigate the biodiversity? We want to investigate biodiversity because we want to document the richness of diversity that our country has. And while doing that, we concentrate mainly on extreme pristine habitats as well as biodiversity hotspots. Whenever our investigation documents, whenever we find any novel species, we document, we report such novel species. If we come across any of the species which are endangered species, we try to come up with the means to conserve these endangered species. We want to explore the industrial applications and we want to prevent the biopiracy. These are the objectives behind investigating the microbial diversity. As you can see, our scientists have covered entire Western Ghat region, which happens to be the biodiversity hotspots, and also the regions in northeastern India as well as Himalayas. You will see that our scientists have been investigating this using the conventional ways as well as using molecular approach. And by doing that, over the period of last 1 year, they have documented 15 novel species of diatoms and one species of angiosperms. Our scientists have also realized that there are several varieties of orchids which can be found in Deccan Plateau as well. And our scientists have come up with the ways or techniques to conserve these endemic orchid species and reintroduce them in the nature. Our scientists are also in taking efforts and they have initiated plans to set up a nursery of orchids which are loved by one and all. While doing that, our scientist also believes that whatever knowledge we gather, that knowledge has to be compiled, that knowledge has to be published and made available. Whatever knowledge we have, that needs to be documented. We can protect our intellectual property rights only when we have documented our property rights, only when we know what resource we have, what wealth we have. As an effort towards that, our scientists have published during this year two books. One was on genus of Caparis in India, and another one is Compendium of Raw Botanical Drugs of Maharashtra. Now, this is a very important contribution as many of the botanical drugs or many of the plants which have medicinal properties, they have not been documented. And since they were not documented, many of the Western countries, private industries have exploited our natural resources. Now, in an effort to prevent that, our scientists have started publishing their books. We also have a repository or culture collections. We have Agharkar Herbarium of MSCS, which holds more than 33,000 herbarium specimens. We also have a biggest diatoms collection, and we have a depository of crude drugs, where more than 3,000 authenticated crude drug samples have been preserved. Many of the industries, when they want to get their crude drug samples authenticated or validated, they always approach us and our scientists help them. One of the groups in Agharkar Research Institute also has been investigating the diversity of fungi and lichens. They want to investigate the diversity, document novel taxa. About the lichens, we have very specialized group because this group is probably one of the rare groups who have the ability to cultivate lichens in the laboratory environment, isolate their metabolites, which have terrific medicinal properties. They have also documented several of the medicinal properties of the lichens and their therapeutic potential in the form of book. And three of the scientists from our institute, Bharti Sharma, Suresh Lomte, he was a research student, and Subhash Gaikwad, they have come together to write this particular book. We also have the largest collection of fungi in India, and the National Fungal Culture Collection of India houses more than 10,000 fungal specimens. We have been giving these fungal specimens or fungal cultures to different academic institute, research institute, and close to 400 research institute and academic institutes have taken benefit of our culture supplies as well as the services such as identification, characterization, culture preservation, etc. over the period of last 1 year. Along with fungi and plants, other group in our institute has also been investigating the diversity of foraminifera. This is a phylum of single cellular animals which are known as protists. And these animals are very significant as they are considered as a proxy for paleo record reconstructions. Usually or conventionally, these foraminifera were identified on the basis of morphological characterization. But as you can see in this picture, they all look similar, but at the same time, we can also identify them as kind of different from each other. Obviously, there is a group of scientists who would call them as the same group, and there will be group of scientists who would oppose them, who would consider them as different organisms. There is always a battle between the lumpers and the splitters. Obviously, the scientists in Agharkar Research Institute, they accepted this as a challenge. They came up with a molecular approach to identify these foraminifera samples, and they started identification. Their identification became unequivocal as it was based on the DNA-based characterization. This kind of contribution was definitely recognized not only in India but internationally as well. Our scientists were invited to be part of IODP, that is International Ocean Discovery Program, as well as they were also invited as a member of the team that visited Antarctica, and from there they collected several such samples. One of the reasons or objective of collecting these samples were to identify the climatic changes that took place. And our scientists, after collecting these samples last year only, they have already started publishing the papers and in the form of books as well as in journals. And one such paper has identified the ocean circulation, impact of ocean circulation in high northern latitudes and its impact on Arctic climate through the ages. This is a very interesting studies that we have been doing, and it's not only about microfossils; it is also about mega or macrofossils. We have a repository where we have more than 5,400 megafossils collection of so many megafossils, as well as 2,000 microfossils, including foraminifera, pollens, and spores. This is third largest fossil collection in India, and it is a unique facility where many people always visit. In addition to the plant, fungi, and animals, we have a group that also investigate the diversity of microorganisms. And this particular group, they are using these microorganisms for applications related to bioenergy. This group has been concentrating on some of the unique organisms. These organisms include anaerobic fungi, anaerobic bacteria, methanogens, and methanotrophs. The objective behind this is we should have the cultures which can degrade the lignocellulosic biomass without any pretreatment and use this biomass for extraction of energy in environment friendly manner or in a benign manner. For that purpose, we depend on anaerobic fungi. We are only one of the two groups working in India on anaerobic fungi. We have very large collection of unique fungi. Some of the novel taxa and genera have been identified as a part of this investigation. We also have a collection of methanotrophs, and these methanotrophs were isolated from various paddy fields in India, and many of these species are novel species. And these methanotrophs are being used for methane mitigation to control the methane in the methane emissions in paddy fields, as well as to use these methanotrophs for the production of some of the specialty chemicals. And this is something that our group has been doing. But one of the most important part that our this particular group has done is regarding the technology for biohydrogen production. This group has developed a microbial process to produce hydrogen from rice straw. We all know rice straw is a burning problem, figuratively and literally at present, especially in northern India. Because of the stubble burning, there has been great pollution. And if this pollution has to be controlled, then you have to take care of rice straw without burning it. What can be done? Farmers do not have any revenue generation from rice straw, and obviously they are left with no other means but to burn it. What we propose is use this rice straw for extraction of energy. Now, if rice straw is to be used for any of the microbial processes, then it has to be pre-treated with alkali as well as heat, and that is going to make this process energy intensive as well as polluting. What our scientists have done is they have come up with a consortium of anaerobic microorganism which can degrade this rice straw without any pre-treatment at ambient temperature and produce hydrogen in significant quantities. The slurry that comes out of this reactor can then be put for biomethanation, and in second stage the methane gas can be generated. This methane gas can be can be reformed into hydrogen. This hydrogen can then be used as a fuel to support the fuel cell based electric vehicles. Now, this is a technology that has been developed for KPIT Technologies Limited. KPIT have already developed a fuel cell. They have shown that they can use this hydrogen fuel cell to run vehicles such as cars and buses. Now, the only problem is what is going to be the source of hydrogen. Our technology, our collaboration is going to lead to generation of decentralized units where hydrogen can be produced in different parts of the country. And this is going to be a very significant contribution. We are keeping our fingers crossed. We have definitely proven the concept. We have the proof of concept in our hand at 50 or 60 liter capacity reactors, and scale up studies are undertaken. We have also developed a process for conversion of rice straw and other agro waste into methane without any pre-treatment. And the yield that we get is very high. We get close to 300 liters of methane per kgs of rice straw, which is close to more than 85% of the theoretical yield. That itself shows how important this particular process is, as since it does not depend on chemical and thermal pretreatment, the cost of the process comes down, the profit margin go up. And as a result, burning of rice straw can be avoided. The rice straw, which is a cost center for the farmers, is definitely going to be a terrific profit making venture or source for the farmers in a near future. There is third area where our scientists have been working, and that is a bioprospecting area, where they have been investigating various plants. By biosec guided fractionation, they isolate the active principles from these plants, and these principles are then used for several medicinal applications. One such application can be considered. I mean, there are different diseases that they target, right from cancer to Alzheimer's disease. I will take one example of Alzheimer's disease, which is a multifactorial brain disorders, which involves events like amyloidogenesis, acetylcholine deficit, neuroinflammation, cellular oxidative stress, and dysfunctional autotrophy. Since this is a multifactorial disease, if at all one has to cure the Alzheimer's disease, it is necessary to target multiple mechanisms at the same time. Our scientists have proven that the extract of Crinum woodroffe plant has been showing some effect against the Alzheimer's disease in the laboratory model. They have purified the active principle from them, and this particular compound, galantamine, has been shown to be very effective as it can show the anti-inflammatory activity as well as acetylcholinesterase inhibitory activity. What our scientists have done is they have started modifying such active principles which were purified, come up with the derivatives which are less toxic or non-toxic, which are more active. And one such derivative has been the acetylcoumarine phenylthiosemicarbazole derivatives. And these particular derivatives have shown significant acetylcholinesterase activity, anti-inflammatory activity, anti-oxidative activities. They also have a favorable effect on autophagy. All these mechanisms are very important as they help in controlling or containing the development of disease such as Alzheimer's disease. And that is the reason why this particular compound is definitely considered as one of the most significant achievements. However, these are early stage, and our scientists are going ahead with some of the more advanced studies to come up with something concrete that can be called as a medicine for the treatment. The developmental biology group is one of the groups in our institute where they concentrate mainly on the basic research. As almost all other groups are concentrating on applications, this particular group has been has been carrying out research, basic research to understand the mechanism regulating cellular signaling in development and diseases. And they have been using different models such as zebrafish, they have been using Drosophila model, they have been using the Hydra model as well. I would like to take a couple of such example to highlight their finding. One such model is Hydra model, where as we are aware that most of the cells, they also have at one time or other damage to their DNA. And when there is a damage to DNA, obviously all the living cells also have the repair mechanism. And because of such repair mechanism, such as nucleotide excision repair pathway, one can repair the damage and be cool about it. Now, in Hydra, one such compound which is known to be a very important molecule in nucleotide excision repair pathways is xeroderma pigmentosum. And there are like several such proteins right from A to G of this particular protein which are available and which play very important role. Now you have human system at one hand and you have hydra system at another hand, which is a primitive system which was developed millions of years ago. But the best part about this particular animal is it has a regenerative capacity. In this particular hydra animal, when the DNA mapping was done, it was observed that it had a gene which encodes for XPA, and this XPA is a homologue of human XPA. When they investigated the nucleotide sequence, they found out that there was 58% similarity. But when they studied the amino acid level homology, they found out that homology was more than 90%. When their structures were superimposed, one can see that there is a great deal of overlap. Now, this clearly indicated that Hydra had this particular protein. Definitely, it was of interest that how homologous it is to the human in terms of functionality. Can it repair the UV-induced DNA damage in human systems also? And such studies were carried out, and in such studies, it was found out that human cells in the cell line studies, when it is damaged with UV-induced mutations, the cells, if at all they are deficient in XPA, they become they become damaged. They can be easily killed. However, if at all you support them with human XPA, more than 90% of these cells recover. Now, what happens if you supplement them instead of human XPA with Hydra XPA? You will see more than 50% of the cells recovering. Now, that clearly indicates that functionally also Hydra XPA is complementing human XPA. The low level of the repair could be explained by its inability to bind to replication protein A70, and which probably is causing, since the hydra molecule is not binding very efficiently with this, there could be limited DNA repair. However, this is a very important observation that uh, which is considered as significant because it gives us insight into evolution of the DNA repair repertoire which originated millions of years ago. Another scientist in the same group has come up with another protein, and this particular protein is considered as very important because whenever there is any damage to heart at a time of heart attack or myocardial infarction, which results in the damaged muscles in heart. As a result, that particular part of the heart becomes non-functional or kind of useless. What our scientist has been able to show is, if at all you use the CCN2A protein, then it can help in regeneration of the heart in zebrafish model. This is a very important clue that he has got, and obviously several studies are being carried out to explore these findings further. There are several such interesting findings from that group, but I will not go into details of that due to time limitations. We have genetics and plant breeding group, which is a fantastic group which is contributing to the betterment of the society by coming up with different varieties of wheat and soybean. Now they are using conventional crop breeding technologies, and they are using advanced molecular techniques to select the desired traits and come up with such seeds which can be resistant to drought, which can require less water irrigation, which are resistant to different pest infection, which require less amount of fertilizers. And these varieties, some of these varieties are also biofortified varieties, wherein there is additional concentration of essential nutrients such as iron, zinc, and protein. So as a result, there are several interesting varieties that our scientists have developed, and 12 varieties of wheat, 11 varieties of soybean, and one variety of grape have been released by ICAR. Now, when they are doing this, they are also contributing to the production of breeder seeds under the ICAR sponsored project. And these breeder seeds are shared with the farmers to again propagate them in the fields as well as to use them to cultivate certain crops. And it has been seen that the contribution of MSCS developed seeds in farming sector has crossed the economy of more than 2,000 crores of rupees. Also, our scientists have been taking very conscious effort to disseminate the knowledge that has been developed with the farmers. They directly go and educate the farmers by conducting the farmer mela, by conducting the WhatsApp group, and also conducting the field demonstration in the form of frontline demonstrations. Interestingly, one biofortified variety, MSCS 4058, was released by Honorable Prime Minister Narendra Modi ji when on 16th of October 2020, on the occasion of 75th anniversary of Food and Agricultural Organization. And this is a variety which is biofortified variety, and the Niti Aayog has identified as one of the thrust areas in the form of to make India Kupushan Mukt Bharat, that is to alleviate the problem of malnutrition. And this variety, where the wheat flour will definitely contain higher concentrations of proteins and essential minerals such as zinc and iron, is going to be ARI's contribution to that particular effort. The last thematic area, certainly not the least, is nanobiosciences, where multidisciplinary research is carried out by the scientists in the field of biotechnology, microbiological, physics and electronics, pharmaceutical science, and virology as well. Many of these projects are sponsored. During the course of last 1 year, this particular group came up with a kit. This kit is for the detection of white spot syndrome virus. Now, this virus is known to infect prawns, and aquaculture is a big business or industry in India. Unfortunately, whenever there is any infection of white spot syndrome, that can be diagnosed by molecular techniques such as PCR, meaning that farmers cannot do anything on their field. They will have to take these cultures, and these cultures will have to be tested in the NABL accredited laboratories hundreds of miles away from the farms. Many times, if the testing is not adequately done, then many of these consignments which are exported to foreign countries are rejected. What our scientists have done is come up with the 10 minutes assay, which is a lateral flow based assay, which can detect this virus within 10 minutes, and this detection can be done on site. This is a extremely important contribution, and these scientists have not only concentrated only on WSSV virus, but they have also come up with the detection kit for the Macrobrachium rosenbergi nodavirus as well as myonecrosis nodavirus. And these are very important diseases, and it is mandatory that whenever any of the marine consignment is to be exported, one has to carry out the certificate for detection of these viruses. Another important contribution that came from this particular group was the hemostatic zero gel dressing. Now we know that bleeding can be very profuse when there is an accident or when there is an injury. And as a result, many times if this bleeding is excessive, then it can prove fatal as well. What our scientists have done is they have come up with a specially treated bandage or dressing. Now, this dressing can interact with the RBCs, they can activate the platelets and prevent the bleeding very fast. Not only that, it results in the formation of the stable clot. And as a result, after 2-3 minutes, even if one has to remove that bandage, it can be done very easily without initiating bleeding again. Our people, our scientists are working in collaboration with the industry. They have already secured a ATGC grant from DBT, and very soon we hope to commercialize this particular activity. Also, another important part is this particular group has come up with a rapid detection kit for detection of SARS-CoV-2 virus. The beauty of this particular kit is it detects the nucleic acid, that is RNA, and not the protein. As a result, this particular method is more conclusive. The false positive or negative results are minimum, and it is rapid. It can be performed on site as well. The the beauty of this is it can be performed by any layman as well on site by collecting the sample and doing some kind of pre-treatment. This is a very important finding and we hope to commercialize soon. So just in the end, I would like to take a look at the performance indicator of our institute. Our scientists, which are 32 in number, have secured 69 extramural projects and 9 industry-sponsored projects. They have published 79 publications in the SCI journals and 25 publications are in non-SCI journals. We have been steadily making progress towards filing the patents. We have also transferred 3 technologies to SKR Agrotech Wardha. These are the technologies developed by Nano Biosciences Group. We have academic collaborations in India as well as abroad. There are 37 different academic institutes with whom we have collaborations in India and 15 institutes abroad. We have PhD students and they are the pillar of our research. We have 59 students who are registered at present and 8 students were awarded PhD degree over the period of last 1 year. Our scientists have been sharing their knowledge as a societal responsibility with the farmers, with the common man. And what they do is because they share them the good crop cultivation practices, they share with them the breeder seeds, etc. And as you can see, many a times the MSCS varieties they give even higher yield in the fields than what we claim or what we get on ARI field. That I believe is the greatest validation that one could have for the varieties that developed by our scientists. And I'm very proud to say that 2 of the farmers they secured Maharashtra State Government Chief Minister's Award for getting the highest yield of the crop using the seeds which were developed by MSCS. We have different repositories such as fungal culture collection, microbial culture collection, and other repositories which have been a great source of revenue generation. But more importantly, we have been catering to the need of academic and research institute in our country and sometimes abroad as well. We also have the paleobiology group and where they have been developing which has been done. Also, I would like to say, which is one last important activity, and this last important activity has been the laboratory that ARI has developed. This is a ICMR approved laboratory for COVID-19 testing. And this is very important finding. Till date, our scientists have tested more than 45,000 samples. And these samples were tested, and I'm very proud to say that very recently in the meeting of the directors, DST told us that when the PMO, that is Prime Minister's office, took the overall review of this testing, it was mentioned by ICMR that ARI happens to be the only laboratory from where there has not been a single false positive or false negative result reported. And that speaks volume about the contribution of our scientists, their dedication, their expertise, and their methodical approach in which they have been doing this particular testing. We have taken conscious efforts to publish our findings in the form of popular article in various vernacular languages also. There has been excellent media coverage for our finding. And lastly, one of our scientists, Dr. Monali Rahalkar, has been acting as a crusader to find out the origin of SARS-CoV-2 virus. And she has been doing a lot of work along with the help from her husband, Dr. Bahulikar. And both of them, they have published a lot of reports and their findings, their publications were noted by international media also. She has given several interviews in different international media as well. And this particular finding is very significant as it was able to trace the origin of SARS-CoV-2 virus to Wuhan. More importantly, I would just like to touch upon the future plan when our scientists in this, after completing 75 years, which is an important milestone, they definitely come up with a new 5 years vision. they need to improve the patent filing and IPR protection exercise, which all of us have taken as a challenge. And they are going to try their best to transfer more number of technology, increase the industrial collaboration, improve the knowledge dissemination further, and contribute to human resource development significantly. We want to increase our revenue as much as possible and share this with Government of India. And thank you all for your patient listening. And I must mention in the end that our members of our governing body who have been pillar of support for us, life members of MSCS, who have been pillar of support for us, without their guidance, without their vision, it would not have been possible. At the same time, terrific efforts which have been taken by our scientists to come up with different research projects and come up with different findings is really noteworthy, and I am in great depth of their contribution. I also mentioned the contribution done by the administrative and technical staff of Agharkar Research Institute, who have facilitated the smooth functioning of this institute. Thank you so much.
Thank you very much, Director Sir, for your report. We could hear in nutshell the entire activities of ARI for this year, and it was very, very interesting. Thank you again, sir. So today we have among us Professor Sandeep Verma, Secretary SERB, and he will be delivering the 61st Professor S.P. Agharkar Memorial Oration. Professor Sandeep Verma is currently serving as Secretary SERB, New Delhi, and he is on leave from Indian Institute of Technology, Kanpur. He obtained his PhD from University of Illinois Medical Center, Chicago, and subsequently held postdoctoral position at Johns Hopkins University Medical Institutions and Max Planck Institute of Experimental Medicine. He is also an adjunct professor of biological sciences and bioengineering and affiliated faculty at Center of Nanoscience and Center of Environmental Science and Engineering, IIT Kanpur. His research interests are in the area of chemical neuroscience, antimicrobial resistance, bio-inspired nanomaterials, and platform detection technologies. He is a recipient of the Shanti Swarup Bhatnagar Prize, the Goyal Prize, the Swarna Jayanti Fellowship, DAE SRC Outstanding Investigator Award, and the Distinguished Alumnus Award of Banaras Hindu University. He is an elected fellow of the Indian National Academy, Science Academy, Indian Academy of Sciences, National Academy of Sciences India, and the Indian National Academy of Engineering. He is an associate editor of the Chemical Communications publication from RSC UK and also on the editorial advisory board of ChemBiochem and Journal of Peptide Science, which is a Wiley publication. We have such an eminent personality amongst us, and now I invite Professor Verma to kindly give his oration.
Namaskar. Good morning, everyone. Thanks for your words of introduction, madam. Let me start by thanking Dr. Dhake for this kind invitation. It is a delight, a privilege, and a true honor to be with all of you and to deliver the 61st Professor Agharkar Memorial Lecture. I am truly honored, and thank you so very much for this opportunity. So, if you would please allow me, I'll like to share my screen, and please let me know when you are able to see it. It says I can't share my screen. Am I missing something?
We'll just check at our end, sir. Can you screen share it once more?
It says can't share your screen.
Okay, just one.
Sir, can you please try now?
Okay.
Sure.
It's not allowing. I am using Firefox. Would there be a problem using Firefox?
We made you co-host. Sir, is it possible to
Should I use
Chrome, please?
Chrome?
Yeah, yeah. Please, sir.
So I would apologize and maybe log out for a minute or so. Huh?
Sure, no problem.
Okay. Thank you.
So we will just wait for Professor Verma to rejoin.
Till Professor Verma joins, I would like to share with everyone that our guest of honor, Professor Karmalkar, will not be able to join us today as they have unexpected visit by minister without any announcement. So he is kind of occupied there. So in his place, Dr. M.C. Srinivasan will be our guest of honor. I request M.C. Srinivasan sir to please accept our invitation to be the guest of honor, sir.
Thank you so much, Prashant, for this honor that you are giving me on this memorable occasion. So I will definitely sort of do whatever I can in my capacity as the chief guest today at this function. Thank you very much.
Thank you so much, sir. I appreciate it.
So he's not coming. He's not coming.
Yes.
Hello. Are you able to see it now?
Yes, sir.
Yes.
Yes.
We are able to see it.
So please tell me if it is full screen.
Yeah.
Yes.
Yes, sir, it is.
Oh, I'm so sorry. I mean, this should not have been. This should not have happened. I apologize.
No problem,
So
sir.
no problem at all.
Thank you very much again, Director Sir. I would really like to thank all who have joined us this morning for my lecture, which is going to be based around peptide-centered strategies in chemical neuroscience and stem cell engineering. And as it was pointed out, I am currently at SERB, but my primary affiliation remains IIT Kanpur, where bulk of this work has been done. And I'm very happy to present this work subsequent to the fact that what was presented in the annual report by director pertaining to Alzheimer's disease and the approaches used by some of our colleagues in Agharkar Research Institute to alleviate Alzheimer's disease through a particular strategy. So I think I'm going to add to that field a bit when I talk about chemical neuroscience. But before I go, I'd like to tell all of our listeners or whoever is participating in this lecture today that my group at IIT Kanpur, as this picture shows, works on three major verticals. So if you start from left hand side, we are looking at a lot of protein assemblies, protein based systems that have relevance in diseases, in neurological diseases in particular. If you look at the center, we are showing you a lotus leaf and this super hydrophobic surface where water droplets are assembled. So we are also interested in customized surfaces which have a particular application. And I'm not going to talk about that today, but we are also interested in the rightmost corner. You see a lot of batteries. So we are working on nucleic acid based, at least for us, very exciting. And I'm going to showcase you just a part of what we do in these areas. So before we start further and go more into science, the acknowledgements are due to my PhD students. I would not read the name. They are all listed here. Some of the recent alumni who are at the bottom. There are postdoctoral fellows. There are a number of collaborators who work with us on these issues, these research problems. And of course, funding comes from a variety of government funding sources, which includes Department of Biotechnology, SCRB, and DST Nanomission. So the first aspect of our talk today would deal with what is chemical neuroscience. And what I would like to sort of share with you is that how chemistry or chemical sciences, chemical structure-based strategies would bring us value when you look at solving a particular problem in neuroscience, such as Alzheimer's disease or Parkinson's disease, or something even more fundamental, which is release of a gas inside a neuron. So this is what we specialize in. And uh, hello, are you able to see it?
We
Yes.
are able to see the slide.
Oh, okay. So we are going to talk to you, or I'm going to tell you about a very simple molecule, which is gaseous molecule, that is nitric oxide. And this nitric oxide gas, or a small molecule, it has very interesting properties. We know nitric oxide for very many reasons in small molecule chemistry. But if you look at biochemical aspects, nitric oxide serves as a very important neuromodulator and neuroprotective agent. The idea here is that if you can generate nitric oxide in just the right amount, it has the definite property of neuroprotection. But if this nitric oxide is generated in higher amount and it goes and does harm to very many pathways within our neurons, then it exhibits what is neurotoxicity. So it is an important molecule for, as I mentioned, intracellular neuronal signaling, regulation of neuronal metabolism, and a variety of biological activities and biochemical pathways within the confines of a neuronal cell. So a natural question is that if it is a gas, how do we generate this gaseous molecule inside the neurons or inside the cells? So basically, you would go through this particular starting material, which is L-arginine, and L-arginine, which is which is an essential amino acid, is acted upon by nitric oxide synthase enzymes, and L-arginine gets converted to citrulline, and this conversion releases nitric oxide gas. All right, so nitric oxide is released, which can go inside the mitochondria, interfere with quite a number of mitochondrial functions. It can go and do what is called as S-nitrosylation. So the thiol groups, SH groups, get nitrosylated, or it can also nitrosylate tyrosine, which is again another essential amino acid. It gets nitrosylated to give you what is called as nitrotyrosine. So synthesis of these species would offer you neurotoxicity if it is in large target cell through diffusion process. Right now, you do not have a molecule that is going to be solid in nature or dissolved in the fluids, but it is a gaseous molecule. So what it does, it just diffuses through the cell boundaries, and it has a property to activate what is called a soluble guanylate cyclase. Soluble guanylate cyclase is an essential enzyme that converts guanosine triphosphate to cyclic guanosine monophosphate, right? So this second messenger, which is cyclic guanosine monophosphate, has again a variety of activities, one of which is important for us, that is synaptic plasticity. So it works on neurons. It also smooths muscle relaxation. So a number of physiological manifestations by this small molecule. And I can go on and on, but for the paucity of time, I hope I have impressed upon you that this small molecule has a number of activities which depend entirely on the amount of nitric oxide which is synthesized, right? So a natural question one could ask is that if it is a gas, because we know if we want to eat aspirin, we take a tablet. If we want an injection, we take an intravenous fluid-like system that is injected in us. But if you want to deliver a gas, how do you deliver a gas inside a molecule or inside a human being, and so on and so forth? And for this, the big question is of delivery. I would not go into the detail, but I would like to sort of point out that long back there was such a molecule reported in literature where this organic nitrate, if you will, or nitrite, was used to deliver nitric oxide, and other kind of activities were studied, not the studies that we are focusing on. So we thought that it's a very cool strategy that if we can have nitrite, we could deliver nitric oxide gas through a different mechanism inside the cell. We can control its delivery. We can control the concentration to which we want nitric oxide is to be synthesized over a period of time. So sustained delivery of a gas inside a neuron for a prolonged period. So it's a very challenging task, and I'm very happy to tell you that we came up with a molecular design. So this is a molecular design from scratch. And if you would look at individual components of this molecular design, I would like to sort of walk you through this particular molecule. If you can see my arrow, this is your classic salicylic acid molecule, right? Salicylic acid, which is core of dispirin or aspirin, was taken, and the entire molecule was built around salicylic acid, where the carboxylic group was connected to a dipeptide. Ditryptophan, mean tryptophan dipeptide, was connected to the carboxylic acid, and the phenolic group was nitrated the way it is shown here. So we are putting this dipeptide for self-assembly, and this organic nitrate or nitrite, which is nitrite, which is going to be the trigger to release nitrite anion inside the cells, followed by generation of nitric oxide. So looking at the dipeptide assembly, one can expect such kind of thing to happen. And if you were to take the organic nitrate part, with the help of its interaction with cellular thiols, we have number of sulfur containing molecules in our body. They are the prime reactants for such type of system, and when they react, they would get themselves nitrosylated, and that nitrosylation would eventually lead to nitric oxide synthesis. So this is the whole contraption in terms of molecular design, and needless to mention, we prepared this molecule. And when I show a successful story, I would at least like to tell all students, and they would agree with me, there are a number of unsuccessful stories. So the design did not arrive at this molecule just like that. We tried at least 30 different molecules to come to a stage which was perfect in molecular design, and we were ready to take it forward and check if indeed it is possible to release nitrite anion inside a neuronal cell culture or otherwise in vitro with the help of a cellular thiol which can challenge our molecule. So the conjugate was taken, which is shown here. The nitrite part is being highlighted because that is going to react with the SH group of glutathione. And most of you know glutathione is endogenous thiol containing molecule. So we will not go into the enzymatic or the numbers of concentrations, etc. But more important for us to realize is that when you react conjugate one with glutathione, you release nitrite anion, and that takes quite a number of successive steps to release nitrite anion, and I've shown it here. So you would have your organic nitrate react with thiol. The nitrate gets transferred to sulfur, then another sulfur molecule attacks on it, you form a disulfide bond, and you release nitrite anion. And once you are able to release nitrite anion, it is reduced in our body by a variety of enzymes, and you would synthesize nitric oxide. So the successive step is you have your molecule, you get it challenged by a thiol, you release nitrite anion, which gets reduced eventually to nitric oxide. So that is the mechanistic pathway we wish to follow. And the subsequent experiments are basically being designed so that our hypothesis is in order and we are indeed getting the results which are due to the release of nitric oxide. So if you look at the profile, the release profile, you would see a beautiful sigmoidal curve which tells you the cooperativity of release of nitrite anion. So this sigmoidal curve, and we will not worry about the concentrations because those semantics can be looked in the publication which is already out. So you look at this sigmoidal curve, there is an inflection point, so you can figure out at what point of time you are actually getting T half and so on and so forth. So we did a number of kinetic calculations to eventually find out that 1 millimolar of glutathione, that thiol containing molecule, was perfect to give us the right kind of release which we are looking at to mimic what concentration we would like to achieve within a neuronal cell, right? So looking at that, now we moved ahead. So from test tube experiments, now we have come to cell culture experiments, and we have chosen Neuro 2A cell line to study the release of nitric oxide in our system. And what I am showing you in the leftmost top leftmost panel is that at zero concentration of our molecule, there is a basal fluorescence being observed. And when you put in 50 micromolar of our molecule of the conjugate one, what you find is that when you put the right kind of dye within a neuronal cell line, you can see enhancement in the fluorescence, which is reflective of the fact that nitric oxide is indeed being synthesized, and the fluorescence that emanates out of the synthesis of nitric oxide is being measured over the basal fluorescence, which is shown here. So there are standard deviations shown here, but students were very happy. I was also quite excited that this molecule is going inside the neuronal cell. The reactions are happening, and eventually nitric oxide is getting released, which is measured by this fluorescence output showed in this particular figure. If you look immediately down, immediately underneath, you would find that from 0 hours to 48 hours, a very beautiful kinetic profile is followed. So you would hit a maxima at around 28 hours, about 28-30 hours. And then when the entire molecule is getting consumed, when 50 micromolar of the molecules are getting consumed, the amount of nitric oxide release would come down. And that is what you expect when you have a drug, you have eaten it inside the body. Then once the metabolism takes place, you would slowly get the concentration out. The concentration goes to a bare minimum level, and which is almost similar to at what you see at 0 hours. So means the entire molecule that you have put in the neuronal cell line has been consumed, has been chewed up by the metabolic processes. And now time had come to see what exactly is going on in neurons once nitric oxide is being released. So the top two lines of the panel are control experiments where we had used only phosphate buffered saline. So there was just salt water being used in neuronal cell culture. But in the bottom two panels, we have our compound either for 1 day, which is 24 hours, or 48 hours. And I will show you a zoomed out image. But I think some of you can notice, if you can follow my pointer or the arrow here, that at some point of time, we found that certain hair-like features were coming out of neuronal cells. And that told us indirectly that something is happening in the neurons, or something is happening in this neuroblastoma cell line, that these hair-like features, which are called as dendrites or neurites, are growing out of the neuronal surface. So if you were to zoom it out, it looks like this. So in control experiment, So in control experiment, if you are at day 1 or day 2, nothing is going on. But if you look at compound 1 at day 1, which is 24 hours, you already see some hair-like features coming out of the neurons. And at the end of day 2, the entire field of vision becomes so complicated that you see quite a number of these dendrites or neurites coming out. And that tells you that you have delivered your molecule, nitric oxide has come out, and this nitric oxide has done something to grow these hair-like features on neurons, which are not visible if you were to treat just with salt water, right? So here is a control, and what is the consequence of putting your conjugate. So we were quite excited, but what we want to do is that what is exactly going on? What is the mechanism with which you would see this protein overexpression or these dendritic or neuritic growth? And for this, we did a number of biochemical experiments. I will show you only 2 for now, where we looked at the cytoskeleton, the neuronal cytoskeleton, because it is known that this particular molecule actin is essential for neuronal polarity, cargo transport, the growth of neurites, the one we saw in the previous slide, these hair-like structures, and a lot is known about these actin filaments, which form. There is some echo. So,
Mr. Partha should switch.
hello.
Mr. Partha should.
So these ring-like filaments of actin are part of neuronal cytoskeleton. So what we did is that we checked if actin is getting overexpressed because of our conjugate. Long story short, what I am going to show you in this particular panel is that if you take our conjugate, you see certain kind of green fluorescence, which is enhanced when compared to pure salt water. If you were just to take nitric oxide like molecule getting in, or if you were to remove nitric oxide and just take the peptide, so in the absence of positive or negative control or a blank control, which is shown here with PBS, you would like to sort of appreciate that in this particular panel A, not only you are looking at the nucleus, but this intense green fluorescence which comes out of this particular dye, which is FITC phalladone. This FITC phalladone is binding to actin, and this binding process gives you the fluorescence which is being measured in panel A. So what it means is that in presence of our molecule, actin expression is enhanced or actin is overexpressed, which is important because more amount of actin is directly correlated to neurite growth. So if you have more actin, you would see more of these hair-like filaments, and that was quite interesting. In the second assay that we did and we reported, we also probed the amount of neuronal nuclear antigen that was synthesized with the help of our molecule. And here we did some really magical experiment where we took our conjugate and we took the nerve growth factor, human nerve growth factor, which is supposed to do the job, which we see here, which is neuronal maturation, growth of neurites, and so on and so forth. So our molecule was very much in sync in terms of overexpression of neuronal nuclear antigen as neuronal growth factor, which told us that we are looking at a molecule which has the potential to regenerate neurons. It has the potential to deliver nitric oxide, and it has the potential to grow neuritic filaments, which were not seen if you were not to use our conjugate and a normal control blank experiment. So these results were super spectacular. We were very happy, and we are continuing on the path of synthesis of nitric oxide, activating compounds. And a lot of it is now part of published material, and a lot of it is part of patent. And we would like to see how far can we take it, including taking such molecules inside an animal. So I'll just quickly change my focus to another gas, another gas which has important ramifications in neuroscience, that is hydrogen sulfide. Of course, most of us know about hydrogen sulfide. I would not go through this uh, biochemical pathway. I mean, anybody can look up how hydrogen sulfide is produced in our body. But more important thing to realize here is that hydrogen sulfide is an extremely important gaseous molecule, which removes reactive oxygen species. So reactive oxygen species like H2O2, right, peroxide radicals, it's peroxide radical anion, all these reactive oxygen species are very bad for our cells, and H2S scavenges them out. It ameliorates the dopaminergic control. So if you look at from Parkinson's disease standpoint, having hydrogen sulfide is a very important thing to have, because it potentiates what is the hippocampal system of our neuronal pathway of our brain. So we started working on hydrogen sulfide because we thought that if we can come up with a strategy to deliver nitric oxide, we can surely come up with a strategy to deliver another gas, which is hydrogen sulfide, right? So we thought that now we not only we will take our experiments to in vitro, to cell culture, but we would also take our experiment inside an animal model. So I hope some of you would appreciate that a lab like mine, which does predominantly chemistry, but we have invested heavily into everything. We have cell culture of our own, we have animal, we connect to people, we connect to collaborators, but we would like to have as many tools as possible in our lab itself, so that the time to reach the goal, time to reach the successful outcome of these projects is minimized, and we deliver the best result in shortest period of time, right? So what we have done here is that we had envisaged that we will feed our hydrogen sulfide molecule producing compound to this particular worm, which is called as C. elegans. We not only bought or collaborated for normal wild type C. elegans, but we also bought one special C. elegans worm, which is Parkinson's disease model. The reason you would use such type of model system is very simple. If you look at a human being, we have a billion of neurons, 1 billion, right, and more. But if you look at such a worm, which is C. elegans, it has only 300 neurons. So with 300 neurons, the study becomes less complicated. It is easy to follow what is going on, and this particular transgenic worm can be used to track upregulation of dopamine production in real time with the help of HPLC and with the help of LCMS. So you could just use liquid chromatography and mass spectroscopy to assess how much dopamine is being synthesized in a Parkinson's disease model. So this is a very exciting model system. So what we did is that when all these things were ready, we went ahead and designed our molecules. Again, to tell all students in particular that these 4 molecules represent about 50 molecules which were synthesized that were not successful. So sometimes you hear the story, there is a lot of sadness hidden behind, because a lot of work was done. But good point is that we have these molecules, and the idea was that this particular oxathiolane, if you can see where I'm rotating my pointer, that is the key molecule that interacts or that gets metabolized by rat liver microsomes in presence of NADPH and oxygen to yield hydrogen sulfide. So if you take such conjugates over a period of time, it's almost like time dependent release of H2S, which is on the y-axis, you would find that all of these molecules were able to produce to produce hydrogen sulfide over a period of time. And I think number 5 gave us the best result, which is about the half-life of about 300 minutes or so, which was just perfect to use these molecules in our animal model. So what we started doing is that we started mixing our compound. So if I can go back, we started mixing our compound with the feed that you give it to the C. elegans. So C. elegans eats bacteria, right? So this particular bacteria, which is OP50, is being fed to these worms. So what you do here is that you to create an artificial environment of high oxidative stress, you start adding extra hydrogen peroxide, H2O2, to the bacterial feed, which is fed to the worm. So now if worm eats bacteria, which is already high in H2O2, the oxidative stress inside the C. elegans becomes much higher. So precisely that is what is shown in this picture, in this graph. Like the first graph, which is OP50, you have some fluorescence, which is basal fluorescence, if you will. If you add H2O2 and measure it out, you see how much excess fluorescence you have, because you have now created an artificial environment with more oxygen species a molecule that releases more oxygenated species. Now, if you come up with one or the other molecule, you would find that number five molecule, which is right here. So number 5 molecule gives you the most suppression of oxidative stress, which is generated by added H2O2, and the fluorescence, which is right here, comes down all the way when you add number 5. So what it means, that number 5, and I'm not showing you that data, that H2S indeed gets generated inside C. elegans. So it gets generated inside C. elegans. It takes care of the oxidative stress, which is artificially being created inside C. elegans by the feed of H2O2, and it brings down to bare minimum level. And that tells us that this particular molecule, not only in the wild type, but also in the challenge strain of our transgenic C. elegans, we are seeing benefits of the release of H2O2, and that is quite interesting and very important. I must tell you, if you are unable to sort of comprehend the importance here. And the second thing we wanted to do is that we wanted to look at the release of dopamine. Excuse me. So dopamine, as you know, is a neurotransmitter, and it is inextricably involved in Parkinson's disease and Parkinson's disease-like symptoms. And the degradation of dopamine, or having lesser concentration of dopamine or dopamine levels, is reflective of the fact that you would get a neurological symptom, because enough dopamine is not present in the right location of the brain, or right location near the neurons, which is supposed to be present according to the homeostasis. So H2S increases the levels of the intracellular glutathione, which decreases oxidative stress. And what we were able to show when we took the transgenic C. elegans, we were able to show that this particular protein, alpha-synuclein, which is which is fused to a yellow fluorescent protein, we were able to show that once we started feeding transgenic C. elegans with our molecule, we found that the amount of alpha-synuclein expressed went up, and the yellow fluorescence was being measured in presence of conjugate number 5, which told us that our molecule has a beneficial role in suppressing the degradation of dopamine, which is reflected in this particular chart. And that is again a very important discovery, that a small molecule has the potential to cure or to get to some levels of comfort for Parkinson's disease like neurons, and which are indeed present in the transgenic C. elegans. And we are taking this study forward. We are trying to deliver H2S in very many ways, and to see if we can get even more beneficial effect and a better data than what we have produced a couple of years back. Just last maybe 5-6 slides on a topic which I wanted to add. We have another 5-7 minutes about stem cell engineering. So what we have done here is that we have taken human stem cells, and stem cells, as you know, they can proliferate, they can differentiate, and proliferation and differentiation are two different processes. Proliferation is when you go through the cell division and you maintain a stem cell pool. That is proliferation, just division, right? Now, when you differentiate, because stem cells also have the propensity to morph into specialized cells. So these specialized cells are now coming from what is called as a differentiation process. And if you were to look at a better picture, here I am showing you what are called as multipotent progenitor stem cells, which have the possibility of to get derived from bone or bone marrow, and they can eventually be isolated not only from bone marrow, but we isolate them. We isolate in our lab actually from umbilical cord blood. So we take umbilical cords which are disposed in hospitals with due medical and ethical clearances. We use it to harvest stem cells, and that is the basis of our experiment that I'm going to show you. And the idea here is that is it possible to have peptide based systems that can come up with the regeneration, differentiation, or rapid proliferation of stem cells. And for this particular study, we have gone totally in the sort of 180-degree opposite to the toolbox that we have in our laboratory. So instead of cell culture, which we have done in the second phase of this work, we started with what we call as stem cell biomechanics. The idea here is that can you take a stem cell and would you be able to calculate what is called as the Young's modulus of stem cells? Means you are measuring the overall fluidity of these stem cells when you apply your peptide or when you apply your sort of small molecule. Basically, small molecule and cell interaction would eventually lead to sort of modifications in tension, compression, and what is called as shear flow microenvironment. And the idea here is to use atomic force microscopy, the AFM indentation that we do in our laboratory. We have our own AFM where we take a highly flexible cantilever because that has to be very effective for cell surfaces, not the hard surfaces, right? And it is possible to create stress versus strain relaxation curves. It is possible to calculate Young's modulus, and by applying something which is called as the Hertz model. So Hertz model, the mathematical equation is shown here, but what we are eventually interested is in determining this F, which is the force constant. Force constant is eventually going to give us an idea what is the balance of stress versus strain, the membrane fluidity, the Young's modulus, that telling us that how we have made our cell more flexible or otherwise. Yeah. So force mapping was done with two different peptides. One was the L form of cysteine, alanine, glycine type peptide. So very simple chemistry. The idea in my lab is chemistry should be tough or simple, it doesn't matter, but the application should be extremely robust, right? So we published it last year. So here we have calculated the stress versus strain curve. We won't go into the detail. Some of you who are interested, we can always talk offline or over email. We found that we were able to induce fluidity in our system. And the Young's modulus was 2.5-fold increase. It increased 2.5-fold in the presence of our peptide. So we were doing the right thing. Then now we were able to go into application part. And I won't tell you the entire story, but one thing I'll tell you is about tissue regeneration, which is coming from a very nice essay, just last 2-3 slides of tissue engineering, where we take stem cells, we culture stem cells in a flat petri dish. Yeah. So in this petri dish culture where you are seeding with a given amount or given number of stem cells, we put an artificial wound, right? So if you have a petri dish, you put a comb, you put something that would be what is called as a wound inducing insert. You let this culture sit for 24 to 48 hours. Once it has cultured enough, you remove this particular wound insert, you remove the insert, and you would find that there is gap in your culture because that part was occupied by the insert. So when a specific part is being occupied by the insert, cells don't grow in that location. So you have a space. And now the idea is if the cells were to divide, cells were to replicate, how quickly can they replicate to fill up your field of vision? Your field of vision gets filled up by proliferation, and that's the experiment. So this is a artificial wound inducing insert followed by proliferation to give you the desired data. And I'm very happy to share with you, if you look at this particular yellow arrows and the broken line, that is your wound, right? So between these two arrows, there is the there is a field of vision where there are no cells, zero cells, right? Now you let them grow as if they were growing just without any external pressure. But if you start adding these tripeptides, you would find that on a certain time scale, having peptide increased the amount of proliferation, and that was quite remarkable. And we were able to connect the fluidity of the cell, the Young's modulus changes in the cell vis-a-vis tissue engineering, or the rate of proliferation of human mesenchymal stem cells. And that was a very nice paper, nice idea we created. And now we have gone ahead, we have done some wonderful work on this idea much ahead, and we are about to publish it in terms of vascular engineering. We are trying to engineer the blood vessels in using the inserts of mesenchymal stem cells. And again, as we had done with our nitric oxide, lot of these biological experiments were done to show that not only fluidity, not only change in the fluidity, but the application of tripeptide is also it also controls protein synthesis, cytoskeletal protein synthesis. And we use a couple of assays, including vimentin and alpha tubulin actin assay, eventually to figure out that indeed these two molecules were overexpressed. If you see this particular lane, the conjugate number 2 over control, the conjugate number 2 has more amount of protein being produced. So that was our contention, that compound 2, which is DCAG. D means the D isomer of cysteine alanine glycine, that was able to give the right kind of fluidity, the right kind of proliferation, which was backed by overexpression of the cytoskeletal proteins, alpha tubulin, actin, and many other biochemical assays that we have done. So finally, I would like to tell you that we work on a number of areas. We are not talking about surfaces and batteries, but we do quite a bit of bioanalytical detection, where we detect. We have even started detecting nitric oxide at parts per billion level, and we have a patent. And if all goes well, we will be able to start a small startup on the issue, or on the function or idea of detecting nitric oxide in breath, in human breath. When you breathe out, we should be able to detect nitric oxide in parts per billion. We also work on new antibiotics. We work on amyloid modulation, as Director Sir was talking about amyloidogenesis, and how semicarbazone, if I'm not wrong, were interfering with the amyloidogenesis process. We do it a lot, in not just by experiments, but we have very strong collaboration in the field of computational modeling of amyloids, and that is where our strength also is, in addition to experiments. And just two papers that have come out in last 2-3 days. So despite despite serving the government, we always get excited when new papers get published, right? So we just published a review in ACS Chemical Biology on short peptides as potent antibacterial and antibiotic adjuvants. And here, as I was telling, that we have now started using the endogenous synthesis of hydrogen sulfide. So we create peptides which sense hydrogen sulfide endogenously inside a cell, and we use it for concurrent bioimaging and for apoptosis induction in cancer cells. And this paper just got accepted in Chemical Science a few days back. So we are looking at very many possibilities in these cutting edge areas of neuroscience and stem cell engineering. And I'm very happy to share some of these results with you this morning. And I would like to thank Dr. Dhakefalkar for this extremely, a very nice opportunity to interact with everyone in Agharkar Research Institute through this Professor Agharkar Memorial Lecture. Thank you so very much. I wish all of you the very best. Have a great day. Jai Hind.
Thank you very much, Professor Verma, for this knowledgeable and informative talk for the 61st SP Agharkar Memorial Oration. And I thank you very much for sharing your exciting and in-depth research for neuronal regeneration, degeneration, and also for tissue engineering. I'm sure a lot of our young colleagues will be inspired by your talk and how you are handling your research as well as administrative jobs. It's truly great to have you online, sir. And thank you very
Thank
much.
you.
Sir,
Thank you.
please do stay with us
Sure.
because the next event is going to be the release of books, 3 exciting books which have been published from ARI. And I thank Dr. MC Srinivasan, who has accepted the invitation for my director, sir, for as the guest of honor. Thank you, Srinivasan, sir. So the next event in our program is the release of 3 books which are published from ARI. So the first book which is published has been edited by Dr. M.C. Srinivasan and Dr. S.K. Singh, which is the practical guidebook to actinomycete biology and technology applications. Dr. M.C. Srinivasan is an eminent mycologist who is an example for several generations of young mycologists here, and he has carried out pioneering work with several species of fungi, and he has worked with isolation and identification of the fungi extensively. And I request Dr. M.C. Srinivasan to enlighten us regarding his publication and give us a few glimpses about the book. Dr. Srinivasan, sir, please.
Thank you, Vandana,
Yes.
for this introduction. Well, I'm a retired senior microbiologist from NCL. I'm presently 86 years old. And I'm a fellow of the Maharashtra Academy of Sciences, life member of MACS, and ex-member of the governing body as well as the Research Advisory Council. So, as I was talking to Dr. Dayapal here, for the last over 60 years, I have been having an association with the Agharkar Institute. So, it is like homecoming for me when I'm talking to all of you here. And to be able to participate in this 75th anniversary of the Agharkar Research Institute is a great experience for me. I will talk about it a little later. Now, this book, about this book, I'll only say, I've been having interaction with Dr. S.K. Singh, who is the head of the National Fungal Culture Collection of India at the Agharkar Institute for several years, and published papers in mycology along with him. The present publication, Practical Guidance, is a joint collaboration between him and myself, and we release it today, dedicating it to the memory of Professor Agharkar on this platinum jubilee of MACS founded by him. I'll give you a little bit details about this book. Actinomycetes are a widely distributed group of microorganisms that have sustained the interest of microbiologists and biotechnologists in their search for novel metabolites. In the words of the renowned biotechnological professor Arnold Demain of the Massachusetts Institute of Technology, actinomycetes are the most helpful group of organisms to mankind in medicine and agriculture. The discovery of the anti-tubercular drug streptomycin from a soil actinomycin earned the Nobel Prize for Professor Selman Waxman at the Rutgers State University, USA in 1952. More recently, in 2015, Professor Satoshi Omura of Japan was given the Nobel Prize for his discovery of ivermectin from a streptomyces active against serious diseases caused by pathogenic worms like river blindness and lymphatic filariasis. Recent years have witnessed phenomenal advances in our knowledge and understanding of these organisms. And in the developed world, notably Japan, focus on this group is very strong, and a scholarly society designated Society of Actinomycetologists, Japan, pursue advanced research on actinomycetes and bring out publications twice in a year in their journal named Actinomycetologica. Incidentally, this journal is freely available on the internet, and we have advised our book for the people to sort of refer to this to get advanced knowledge about actinomycetes. Presently in India, contributions to the study of actinomycetes is rather limited. In spite of extensive programs supported in microbiology and biotechnology, there are no active schools of research focusing on the basic and applied aspects of actinomycete research. This needs to be strengthened. India, being a tropical biodiversity hotspot, it can be expected to possess several unique actinomycetes with immense potential for discovering novel and useful metabolites. It becomes necessary to stimulate the interest in actinomycete research among the upcoming generation of microbiologists and biotechnologists of our country. Our present effort in bringing out this practical guidebook for actinomycete biology and technological applications is solely for this purpose. Now, in conclusion, I will say, particularly paying sort of tribute to Agharkar, I like Professor Agharkar's expression, cultivation of science. Like cultivation of a growing crop requires the right inputs of nutrition and environment, the younger generation of scientists need to have the opportunity to absorb and absorb authentic information on the latest developments in the field to build a strong foundation on which these researches could be planned to achieve great success. I hope that our sort of publication sort of meets the requirement of researchers and academicians. I hope and trust that our humble effort to generate interest and enthusiasm in the minds of our young scientists to undertake innovative research projects involving academicians to achieve great success. Thank you very much.
Thank you very much, Dr. Srinivasan, for telling us about your book. It is truly exciting. And thank you, sir, again. And we move on to the next book. As Director Sir said in his report, scientists at ARI are working extensively for soybean cultivation and have compiled this information about crop cultivation practices in the form of a booklet. Dr. Jay Bhai, who is scientist C in the Genetics and Plant Breeding Department, has edited a book in Marathi, सोयाबीनचे सुधारित पद्धतीने लागवड व व्यवस्थापन. So this booklet includes the ideal cultivation practices and methods, such as to deal with the scarcity of water and other conditions during soybean cultivation. So he has included this in a small booklet, which is handy and can be distributed to the farmers during exhibition and other farmers extension program. And they can take advantage of this booklet and follow the ideal conditions which have been suggested. So Dr. Jay has made a small video and we can listen to what he has to say. So I'll be sharing a video with you.
नमस्कार. सोयाबीनची सुधारित पद्धतीने लागवड व व्यवस्थापन या प्रसार पुस्तिकेच्या माध्यमातून आम्ही महाराष्ट्र राज्यातील सोयाबीन उत्पादक शेतकरी बांधवांसाठी सोयाबीन पिकाचे संपूर्ण शास्त्रोक्त व्यवस्थापन विषयक मार्गदर्शन खिशात मामी येथे आकाराच्या पुस्तिकेच्या रूपाने दिले केलेले आहे. या पुस्तिकेद्वारे आम्ही शेतकरी बांधवांना सोयाबीनसाठी आवश्यक असणारी जमीन, हवामान, सुधारित पाण्याची निवड यांपासून थेट सोयाबीन बियाण्याच्या साठवण व हाताळणीपर्यंतचे संक्षिप्त ज्ञान देण्याचा प्रयत्न केलेला आहे. या पुस्तिकेची प्रत शेतकरी बांधवांना सोयाबीनच्या बियाण्यासोबत तसेच शेतकरी मेळावा, कृषी प्रदर्शन व प्रक्षेत्र मीटिंग यांच्या दरम्यान वितरित करण्यात येणार आहे. मी या पुस्तिकेची संकल्पना व प्रकाशनाबद्दल आमच्या संस्थेचे संचालक डॉक्टर पी.के. ढाकेफाळकर सर यांचे व ICAR सोयाबीन संशोधन संस्था इंदोर यांचे मनपूर्वक आभार मानतो.
Thank you very much. So he has shared briefly what he has included in the booklet regarding soybean cultivation. The third book on our agenda is The Seed Morphology and of High Value Medicinal Plant. So I will just share the presentation. So the seed morphology of high-valued medicinal plant has been published by Dr. Vinaya Ghate and Mrs. Namrata Gaikwad. Dr. Vinaya Ghate and Namrata Gaikwad Dr. Vinaya Ghate was the eminent scientist who has retired from the botany department, and Mrs. Namrata Gaikwad is currently in the botany department. So they have both published this book, which has documented the seed morphology with beautiful pictures of 123 medicinal plants which are of high value. Their extensive efforts highlight the property of these seeds, and this book will be very useful to researchers, forest officials, and nature lovers. So these are the beautiful pictures which have been included in this book, and it is a documentation which will be useful for the plants which are in and around Maharashtra, and which has been documented and it is available in the herbarium form, which can be referred to in at ARI.
What is the earlier book of?
So we would like to release the book in the presence of the august presence of Chief Guest Professor Sandeep Verma, Secretary Serb DST, and the guest of honor for today, Dr. M.C. Srinivasan, our eminent mycologist. So these are the three books which will be released. We congratulate Dr. M.C. Srinivasan, Dr. Singh, Dr. Jay Bhai, Dr. Vinaya Ghate, and Mrs. Namrata Gaikwad for this exemplary effort and for all the three books. Congratulations to all of them. So thank you very much, Professor Verma sir and Dr. M.C. Srinivasan for being there. So for the online release of these books, and we will move on now to our next event in our program, which is the address, the presidential address. And as Professor Dr. Srinivasan has accepted on short notice this invitation, I will give a brief introduction of M.C. Srinivasan sir, which not mentioned to say he is a well-known figure amongst all ARI goers. He was the previous head of biochemical sciences at National Chemical Laboratory, Pune. He had eminent work in the identification and isolation of several fungal species, which I would like to say one of these species, Benjaminella, I worked on my PhD also. So thank you, sir. He is a biologist and microbiologist with more than 60 years of research experience, and his area of specialization are fungal taxonomy, biodiversity of fungi, on actinomycetes and extremophilic forms, particularly the alkalophiles. He has significantly contributed to the exploration of microbial diversity and their utilization in developing novel enzyme technologies, especially alkaline protease and cellulose-free xylanases. He has worked extensively on the Conidiobolus species, which is an extremely interesting fungal form having shooting spores. And I still remember how excitedly sir would share the petri dish on which you could see the shooting spores on the cover of the petri dish. So it's a very special mycological technique which he has developed, so they can visualize these mystical organisms. So he has been a member of several advisory committees of the Government of India, like DST, DBT, CSR. Sir was elected to the fellowship of the Indian National Academy of Sciences in 1995. He is the recipient of the J.V. Bhatt Eureka Forbes Prize for Excellence in Microbiology. And he has also been the member of World Federation for Culture Collections for several years. Sir has been associated with the Maharashtra Association for the Cultivation of Sciences for several years. He has served as a on the governing body as a member of MACS ARI, and he has been the RSC member for more than 2 to 3 terms. So, sir, we thank you for your continuing and long association with ARI, and I invite you to kindly give your presidential address.
Dr. Agharkar, Professor Sandeep Verma, Vandana, Jastapri, and all the dignitaries who are sort of participating in this get together. This morning when I came here, I never bargained for this. I never knew that I was going to be asked to be the president and give a presidential address. I mean, it is a very honored to sort of be the president for such a function where we are remembering one of the greatest botanists and educationists, Professor Agharkar, and then commemorating the 75th what you call anniversary of this creation. So I think the function has gone very well, and particularly I'm so impressed by Professor Sandeep Verma's lecture, which is full of so much of information. Unfortunately, I don't have enough background to sort of get the full grasp of it, but I can see the amount of thought that has gone into what you call formulating these experiments. I mean, creating sort of animal system, animal models, and to interpret sort of serious diseases like Parkinson's disease and associated with hydrogen sulfide and dopamine. I mean, you see the sort of magnitude of the problem. So this is exactly what everybody should understand. It is doing an experiment is one thing, but then using your brains to formulate a thing is sort of much more important. I think your lecture must really be a sort of source of inspiration for the younger generation of people. Unfortunately, I'm too old to get inspired to do anything more because I'm already 86 years old. I don't expect I can learn anything and then do from that. Maybe I will be born again to sort of get the sort of knowledge from the thing. Thank you very much for your very nice what you call presentation. Dr. Bhalkar, you are sort of summing up of the activities of the MACS is of the Agharkar Institute is really great. It has been sort of, in fact, I have been, as Vadana said, that I have been a member on your panel for a long number of years. But then what is more important is that I say that today, when you have presented the thing, I can see the diversity of activities in which you are involved and also the high standards that you are trying to achieve. The standards always keep on rising beyond our reach, but it is our attempt to keep on trying for it. And you have been doing a wonderful job, and I wish you Godspeed, and I wish you good luck in your future things. So as you had said, I should really look back. Now the rest of the presidential address, I will try to get a glimpse of over the past. I'll walk down memory lane and then see what is it that I remember very strongly about Agharkar Research Institute. The first and most precious thing that I remember is that my meeting Professor Agharkar. In fact, I met him in 1960 when, we are, I can tell you at this point that we are three generations from the same family who have been associated with the Agharkar Institute. My grandfather, Professor M.J. Narasimhan, a very reputed, he was the director of agriculture in Mysore and a reputed mycologist. Then Dr. M.J. a very well-known name, and he was associated with the Agharkar Institute. And I am the third generation, the Father, Son, and the Holy Ghost. I am the third generation of members from the same family who have had interactions with the Agharkar Institute. I remember the time when Agharkar Institute had started in the basement of the law college. And then that was the time when Devdikar was there. And one incident which I really remember was my grandfather used to visit him often and then have a discussion. And I also went with him once. And he was sort of showing him a sort of a variety of wheat, which was supposed to be rust resistant. Which he had got from some foreign country. And then when he had sort of grown the seedling and he had kept it in the windowsill, and that from the top to bottom that was infected by, even that seedling was infected by rust. That was the western strains of Pachydia gravidae. But then here in the Indian strains, even in the seedling stage, it was completely killing it. So that shows biodiversity in the sense that, Dr. Devdikar was a very, very reputed scientist and very strong in his what you call fundamentals. And he was sort of discussing with my grandfather this particular thing. And Professor Agharkar, when he visited the visited Hindustan Antibiotics colony, where I was staying with my maternal uncle, Tirumalisar, at that time. And that was the time I had the occasion to meet him and listen to him. And I was really impressed by the scholarship of him. And at the same time, he was so simple. The simplicity coupled with high level of intellectual scholarship was something which Agharkar had and which all of us should definitely try to emulate and then follow. Now, and I was elected to as a lifetime member of the Agharkar Research of the MACS, and then I interacted with all the directors, Agte, then Rao, and during Rao's time, the National Fungal Culture Collection of India was being sort of finalized, and I had a lot of interaction throughout the period of. The reason why Dr. Singh agreed to sort of co-author this book is because we both have almost a brotherly relationship. We have been sort of working together, and I have visited Agharkar Institute many times. In every one of his annual conferences, I have come and given lectures. So I am not sort of a staff member of Agharkar Institute, but more than that, I have been interacting with these people, and it has been a pleasure working with him on this book. And I really sort of wish to express my thanks to Dr. Singh because at this age, but for his what you call association and taking the responsibility of putting together the data and then sort of getting it edited and all these things. I mean, the sort of, I will say the initiative was from me, but the initiative was entirely from Dr. Singh. So I sort of, I would like all of you to sort of compliment him for the amount of effort that he has put in for working on, for being an author on this thing. And he had not been working much on actinomyces. Of course, I also have very, very marginal knowledge, but we put together the information because for the benefit of the what you call it, younger generation. In fact, in Agharkar's sort of terminology, the use of cultivation of science, again I will talk about that. Cultivation requires that you have to have the right inputs and also the right environment. And the idea is that it cannot be just sort of theoretically discussed. You'll have to work it on it and then put it. And what we have tried to do is up to at least 2015, 2016, we have sort of collected the literature, references to the original papers. And also advise people to sort of read actinomycetology and some of these things which can sort of expand the scope of their knowledge. Today, with the internet facilities and availability of literature just at the click of the mouse, you'll be able to sort of get any information that you want. So it is not necessary to give bulky books, but then that's why I have titled it as guidebook. This is not a sort of textbook, this is a guidebook. So in which we have people are able to sort of get to the references and then they can sort of they can go deeper. The thing is, this is only a sort of like an appetizer. This is not a meal. So this is an appetizer to stimulate the interest in the sort of microbiologist. It's a sad thing that in India, actinomyces research has taken a back seat. So the idea is to sort of bring it at least, if not to the number one seat, at least to the 10th row. So that people can sort of work on this particular thing because they are the most fascinating groups, you see. I got interested in actinomyces because I learned, I was going to Dr. Tirmalcha and he described a new genus Chenia. I mean, in the book we have given, we have dedicated it to both Waxman and Tirmalcha, and a photograph of Tirmalcha and Waxman. I mean, sort of discussing on China at the Hindustan Antibiotics. When Waxman visited the Hindustan Antibiotics for the inauguration of the streptomycin plant, I think it was the year was 1962, if I remember right. So that is how, we want to sort of remember those particular things and inspire the future generations to sort of do meaningful research because we will have to become globally competitive. At the moment, the sort of biotechnology courses have very, very scanty, what you call, coverage of actinomyces. So we hope that this, what we have sort of written in this book, will be sort of, will appeal to the younger generation of students. I don't care if it doesn't appeal to the older people. They are really not going to do much. But the younger generation of microbiologists and biotechnologists should get the inspiration to study this particular thing. And I hope in future better editions of this topic is sort of published and more books come. And then, the field can expand. We need to sort of develop the expertise. Instead of being shallow, we must go deep. And we try to sort of emulate the topmost scientists abroad and see how best we can do that. And I think Professor Verma's lecture will give an insight how to do that. I mean, how exactly you can plan the thing. And I think they should take a leaf out of what he has said and then build up your own career and things like that. So more than this, I have nothing to say. I have just sort of spoken without any preparation. Whatever came to my mind, if some things which I have said is not agreeable, so please pardon me, because this I didn't even have a 1 hour notice for preparing anything. But I hope I have not done a bad job. Thank you very much for this. Thank you.
Thank you, Dr. M.C. Srinivasan. It was, as usual, a delight to listen to you. And I thank you also for kind of making me realize I skipped speaking about Dr. S.K. Singh, my bad. So Dr. S.K. Singh, who is the co-author of the book, is also in charge of and head of the biodiversity and the paleobiology group for all information of all our participants. And he's also the in charge for the National Fungal Culture Collection of India. So thank you, sir, for and I hope any of my lapses you will overlook, because we are reaching to the end of our program. And now I request Dr. Wagh to kindly give the vote, propose the vote of thanks. Dr. Wagh, sir, please.
Thank you.
Thank you.
Thank you so much, sir. Thank you.
Bye-bye.
Good night to all of you.